Hi, everyone. I think we're going to go ahead and get started. So I'm really excited to welcome everybody to the Society for Developmental Biology's newly named Ethel Browne Harvey Postdoctoral Seminar Series. My name is Whitney Edwards. I'm a postdoc at UNC Chapel Hill, and I'm the new postdoc trainee representative on the SDB Board of Directors. We are super excited to highlight the work of our outstanding postdoctoral members through this seminar series. And following this today's talk, I will announce the full list of speakers for the 2021-2022 session. I'll now turn it over to our postdoc moderators, Alys Jarvela from the University of Maryland College Park and Ryan O'Neill from the National Institute of Health.
Thanks, Whitney. Today, Heather Bruce from Marine Biological Laboratory and Eyal Karzbrun from UC Santa Barbara will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please type your questions into the Zoom Q&A box. And additionally, be sure to stay till the very end of the seminar because there will be a special announcement following today's talks. It's my pleasure to introduce today's first speaker, Dr. Heather Bruce. Currently, Heather's working on halotag development at the Marine Biological Lab in Woods Hole in conjunction with Promega. Today, though, she'll be talking about her research on the evolution of novel morphological structures in arthropods that came out of her postdoctoral work with Nipam Patel. Heather has a longstanding interest in the genetic basis of novel structures that goes all the way back to her undergraduate work with Lisa Nagy at the University of Arizona. She continued this interest in her graduate work with Mike Eisen and Nipam Patel at UC Berkeley, where she studied appendage diversity in crustaceans. During her graduate work, she distinguished herself as an NSF Graduate Research Fellow and UC Berkeley Chancellor's Fellow, and she's been actively involved in Woods Hole's famous embryology course. She's nowhere near done with her quest to understand novelty and its origins, and she has great ideas to start her own lab, so definitely keep a lookout for her application if you happen to be on a tenure track search committee. And so I'm going to let Heather take over, and I'll let you know when you're running out of time.
Okay, can everyone hear me?
Yes.
Okay, good. So I'm interested in the origin of novel structures. Novel structures are often defined as structures that are not homologous to any structure in the ancestor, nor to any other structure in the same organism. So novel structures are those where it's not clear what structure in the ancestor it could have evolved from because the ancestor just doesn't have it. So in this sense, novel structures are fascinating because they break our expectations about how evolution happens, because how do you generate something from nothing? They therefore reveal that we are missing something about how evolution works. Understanding how novel structures evolve is also important because novel structures are often key innovations that underpin the success and radiation of major clades. For example, the mollusk shell, the mammalian uterus, and arthropod sticky outy things. So we can understand, if we can understand how novel structures evolve, we can start to understand and maybe even predict these kinds of major evolutionary outcomes. So arthropods have lots of sticky outy things, many of which have been called novel structures, including insect wings and the Daphnia carapace. So this makes arthropods an excellent group in which to study the origin of novel structures. So to understand the origins and evolutionary histories of these novel structures, we need to understand whether they are actually homologous to any existing structures. And to understand those homologies, I found that we need to understand how to align their leg segments. So how to align all arthropod leg segments has been a century-old question because different arthropod groups have different numbers of leg segments. However, the answer to this question turned out to be the key to understanding one of the most well-known novel structures, insect wings. So there were two main theories for the origin of insect wings. The first theory is that wings evolved from body wall plates like those in wingless insects like silverfish. And it's easy to imagine how these plates could become expanded to form insect wings. The second theory is based on the observation that in many insect embryos, the proximal part of the leg is observed to displace and replace the body wall. This proximal leg region now functions as body wall exoskeleton in the adult. So that idea was then elaborated by Kukalova-Peck in 1983. She noticed that crustaceans have lobes on their legs called excites, such as gills or the plates that cover the gills. So she proposed that two ancestral leg segments had become incorporated into the insect body wall, carrying their excites with them to later form insect wings. More recently, molecular researchers found that insect wings and crustacean excites do indeed express the same genes. So that seemed to support the theory that wings evolved from crustacean leg excites. However, an alternative interpretation of this data is that the body wall of insects just co-opted the genetic pathway for crustacean excites in order to make wings in a novel place up on the back. And that would mean that wings are a novel structure that never existed in crustaceans. So since we now know that insects evolved from crustaceans, one way to figure this puzzle out is to align insect legs to crustacean legs. So if insects incorporated two ancestral leg segments into their body wall, then we might expect living crustaceans to have two extra free leg segments compared to insects. So what I did in my PhD work was to compare the loss of function phenotypes of leg patterning genes in the crustacean Parhyele to what was previously published for insects. And I don't have time to go into the details here, but you can read more about that work in the papers that I've cited here. But to summarize all of that work, I discovered that the leg segments of all arthropods can be aligned, and they have a straightforward one-to-one relationship to each other as schematized here. But today, I'm just going to focus on the proximal leg patterning genes here. So one of the ways that I tested the theory that insects incorporated proximal leg segments into the body wall was by looking at the expression of pannier and arucan. Here, pannier is in red, arucan is in green, and dapi is in gray. So the expression patterns of pannier and arucan are strikingly similar between Parhyele and Tribolium. Pannier in red is expressed in the dorsalmost tissue and marks the true body wall. Below that, arucan in green marks the proximal leg segments and is expressed in a dorsal armband next to the pannier domain in a second armband below that. Now, it was thought that this was the entire insect leg, just these 6 leg segments. But since the expression of pannier and arucan in Tribolium and Parhyele is so similar, and since Parhyele has 2 extra proximal leg segments 7 and 8, that must mean that Tribolium also has these 2 proximal leg segments, but they now form the lateral body wall. So notably, the insect wing emerges from leg segment 8 between those 2 armbands of arucan. And in Parhyele, an excite called the tergal plate emerges from this same position. So therefore, the insect wing appears to be homologous to the Parhyele tergal plate. So what seems to have happened is that wings did evolve from an excite on the crustacean leg. That excite became incorporated into the body wall to form body wall plates. And those body wall plates later evolved into insect wings. So if insect wings evolved from multifunctional excites on incorporated leg segments, then maybe other arthropod sticky outy things did too. And so in order to begin exploring that idea, I decided to investigate the Daphnia carapace. So the Daphnia carapace is a bivalve shell that surrounds the animal like a cape. The carapace forms a brood pouch for the embryos in the space between the cape and the mother's back. So what gave me the idea to investigate the Daphnia carapace was this preprint from Shiga et al. In 2017. They found that the carapace expresses and requires excite genes like vestigial and scalloped and wingless, which suggested that the carapace is an excite, just like the insect wing and the Parhaeli turgle plate. In addition, they found that the carapace emerges from the dorsal head at approximately the maxilla 2 head segment. So the mouth parts on the head are highly modified legs, but they're actually still capable of forming excites in other crustaceans. So I therefore hypothesized that the Daphnia carapace is an excite on the maxilla 2 mouth part of the head, and that it emerges from the proximal most leg segment 8 that was incorporated into the head capsule of the body wall of Daphnia. So we know that Pannier marks the body wall, and Arakan marks proximal leg segments, even if they've been incorporated into the body wall. So this is what I would expect if the Daphnia carapace is indeed an expanded excite on the maxilla 2. So if the carapace is an excite, then Pannier should be expressed in a narrow dorsal domain, and Arakan should be expressed into the carapace. Alternatively, if the Daphnia carapace is a dorsal non-leg structure, then Pannier expression should be expanded into the carapace, and those 2 Arakan armbands should be below or ventral to the carapace. So what we see is consistent with the carapace being an excite, not a dorsal non-leg structure. So in all of these images, the head is to the right. The Parhaeli image on the left is in lateral view, and the 2 Daphnia images on the right are a dorsal view. So in this top image of a Daphnia embryo, vestigial in light blue marks the edge of the carapace, which extends about halfway down the back. And in this bottom image of Daphnia, the carapace is fully extended. So you can see that Pannier in red is expressed in a narrow dorsal stripe, and Arakan expression is expanded into the carapace. And so since that expanded Arakan expression in the Daphnia carapace, that's right next to the Pannier domain, that likely represents the dorsalmost armband of Arakan that we see in Parhaeli and Tribolium. So it looks like the Daphnia carapace is indeed an excite on the proximal 8th leg segment of the maxilla 2 mouth part on the head. So interestingly, other arthropods have that same vestigial positive excite field at the maxilla 2 position that in Daphnia forms the carapace. In both Tribolium and Parhaeli, vestigial in blue is expressed in the maxilla 2 region, nestled between the 2 Arakan armbands, just like it is in the insect wing and the Parhaeli plate, and the Daphnia carapace. Furthermore, in Parhaeli, the maxilla 2 region is extended into a flange, and this flange is reduced when vestigial is knocked out. So it looks like this excite field on maxilla 2 that in Daphnia forms the carapace, that excite field still exists in Parhaeli and Tribolium, even though those arthropods don't form a carapace. And similarly, while Daphnia does not form turgle plates like Parhaeli, these excite fields still appear to exist in Daphnia because the excite patterning genes wingless and vestigial are expressed in segmentally repeating dorsal domains. So therefore, even though Daphnia doesn't form obvious turgle plates, it still retains the regulatory architecture for making them in serially homologous locations in each body segment. And notably, those dorsal fields could presumably be elaborated into large structures in future lineages. So what I think is going on here is that there are serially homologous excite fields on all leg segments in all arthropods. It's just that most of them aren't obvious. So I think this might explain the origin of most arthropod sticky outy things on the lateral body wall. So my thinking here is that these developmental fields can persist in a cryptic, unrecognizable form in intermediate lineages like the silverfish body plates and the parhaeli head flange. And then these cryptic fields can become elaborated again in later lineages, such that they are no longer recognizable as the ancestral structure and give the appearance of just popping out of nowhere, so like insect wings in the Daphnia carapace. So in this model, structures would not have to be continuously present in a morphologically obvious state from ancestor to descendant in order to be homologous. So rather than de novo co-options, these developmental fields are always there, persisting in a dormant, truncated, or highly modified state, and then de-repressed in various lineages to form what look like novel structures. So cryptic persistence of developmental fields could provide a more mechanistically satisfying explanation for the origin of novel structures, in contrast to the current theory of repeated gene co-option. So now I'll discuss some of the implications of this work. And I want to let everyone know that I'm not trying to rile anyone up. I'm just hoping that we can start a discussion about this. So for decades, researchers have been finding that structures on the legs and body wall of insects express the same genes, and these have been called the wing gene network. These were often interpreted to be wing serial homologs and interpreted as evidence of gene co-option. So this has contributed to a widespread narrative that co-option is a common mechanism for the generation of novel structures. However, all of these insect structures may just be excite fields that were inherited from their crustacean ancestors. Thus, the lack of a molecular developmental model representative of the ancestral crustacean group gave the appearance that insect structures had repeatedly arisen de novo, and spurring a mistaken foundation for one of the most widely discussed notions in Evo-Devo. So I think this is a clarion call for diversity of research model organisms in every major clade, so that structures no longer appear to be novel just because the ancestral state hasn't been investigated yet. So a second reason for the dominance of the gene co-option model in explaining the origin of novel structures might be the poor naming of master regulators. So it's often proposed that a novel structure came about when an entire genetic pathway was co-opted. The underlying assumption here seems to be that this structure and its genetic pathway was not present at that location before, and that the tissue doing the co-opting is unrelated to the tissue it is co-opting from, meaning they are different developmental fields. So the related assumption seems to be that a single master regulator can recruit all of the transcription factors necessary to generate a structure outside of that master regulator's developmental field in a tissue not previously primed with dozens of other transcription factors. For example, the idea that dorsal non-leg tissue co-opted leg excite genes to form wings on the back. So the more I think about kind of conceptual terms like co-option or cross-wiring, when people talk about the origin of novel structures, when I work it out, it just seems a little bit less plausible. So in some cases, maybe a better model of novelty and where it comes from is based on what we know about Hox genes. And so for Hox phenotypes, we generally interpret the phenotypes as transformations between serially homologous structures and locations. Therefore, novel structures might just be de-repressed structures at previously unrecognized serially homologous locations. And so that's actually pretty exciting, because then novel structure locations should actually be predictable. They would be revealed just by doing Hawkes transformation experiments and other kinds of experiments that cause transformation of tissue. So I kind of wonder if maybe we shouldn't be invoking gene co-option as often as we do when we're trying to explain the origin of, structures that appear novel. And I think there's a real need to develop criteria for distinguishing between de-repressed serial homologs and truly novel structures. And I'm developing this seminar to address this with Ken McKenna in the Cooper lab. And that's it. And I'd like to thank everyone in the Patel lab, and I'll take questions now. Should I stop my share?
You can probably leave it up.
Okay, I'll leave it up.
Thank you for that fascinating talk. And you actually have a little bit of extra time. I don't see any questions yet, so I will start off. It occurs to me that you're sort of fortunate in that you're studying structures that are hard and easily preserved. To what extent are you able to use the fossil record to confirm what you think about the ancestral state, to see if there are other points in time where these excites might have popped out, and sort of back up what you're seeing with your molecular work?
Yeah, so we do see structures that look like excites in very early fossil arthropods. And so previously it was thought, at least from everything I've read, that excites evolved in crustaceans. And so, but it seems like there are a lot of excite-like structures outside of crustaceans, like in chelicerates, like spiders or like millipedes and stuff. And then all of these fossil arthropods that evolved before, all of the like living arthropods. And so I think, we can use the fossil data to, look for these excites, even deeper in time, and try and piece together what could be excites and what they may have evolved into in all of these different lineages.
Thanks. Okay, it looks like we're starting to get some questions in. So Mark McManaman asked if you consider the excite field to be a morphogenetic field.
Yeah, sorry, I just I tried to like not use a lot of jargon. And since I was already using excite as a jargon thing, I was like, I'm gonna do like developmental field. But yeah, morphogenetic field. Yeah.
Okay, we have another question from Craig Jacobs. It says, very interesting talk. Do you think a lot of the issues occur due to morphological characterization of tissues, e.g. the proximal leg segments being part of the body wall? It looks and behaves like a body wall, but then the gene expression is suggesting otherwise.
Yeah. We don't have, for so many arthropod species, all we have is morphology. Like we don't have gene expression and stuff. We have some embryology, so we can see, in the embryo that like these little things that look like leg segments on the proximal leg, they like kind of moosh and like fuse into the body wall. So we do have that kind of evidence too. What was the second part of the question?
The fact that it looks and behaves like a body wall, but then it's the gene expression data that's telling you that that's not true.
Yeah. So I'm really hopeful for, people working in all other arthropods, if they can look for like pinear expression and aracan expression, and then, if they have a like weird gland or a sticky outy thing on like, the kind of lateral body wall of whatever arthropod they're working on. And if they look for pinear and aracan, and, if it looks like aracan brackets your little structure, then, that it might be an indication that that thing is an excite.
Okay, we have more questions coming in. Bhavna Sawal asks, hi, interesting talk. Why would these excites exist in species that do not have these novel structures? Do they serve a function other than being a platform for evolution of novel structures?
Yeah. So in crustaceans, crustaceans have like tons of excites, and they're like gills. They do like they're like salt glands for like osmoregulation. And they make like plates for like armor to like kind of protect the body wall. In Parhyele, they do, they kind of move towards the center, and then they make like little petals that overlay each other and make a little brood pouch in the female to like hold embryos. So they do like a whole bunch of things. So I think it's just that, you know, those developmental morphogenetic fields, they're still kind of persisting and existing in, you know, intermediate lineages, but then in later lineages like insects or something, you elaborate that excite field again to make wings or something. So wings should just really be considered crustacean excites.
Okay, we have some more questions. Xavier Zolnay asks, as an anatomist, I'm suspicious that the same ancestral number of podomeres might persist through deep evolutionary time. For example, modified legs in millipedes might sometimes completely obliterate many of the more distal podomeres. What are your thoughts? Also, he says that he loves your term sticky outy thing.
It's like descriptive and it's not jargony. Yeah, I mean, so you can tell, you know, ancestral leg segments. You know, you can look at muscle patterns. So, you know, a true leg segment should have muscles like originating in it and stuff and attaching to it. And yeah, so for like millipedes and stuff, when you, this is where the embryology comes in handy. So in like Teague's 1940, who was looking in symphylids, he noticed that the, you know, the proximal part of the leg, which he called the leg base or the limb base, that like, which also carries the spherical, if you're into myriapods, I don't know. So that he observed that to kind of fuse into the body wall. And so that's kind of the same situation that we see in insects where like the proximal, I don't know if you can see where I'm like pointing to me, like the proximal part of the leg just kind of like fuses and becomes the body wall. So that's the kind of, you know, we don't only have to rely on morphology. We can use morphology and like muscle structure and embryology and gene expression to kind of, you know, build like a synthesis or a consensus of like what seems to be going on. Yeah.
Okay, we have another question from Carlos Olivia. Very nice. What do you think is the cellular mechanism for the fusion of this structure with the body wall? Some change in cell adhesion molecules?
Yeah, I don't know. Your guess is as good as mine. But I do think that, I don't know about like at the cellular level, but from kind of a functional standpoint, like, you know, why would you fuse your proximal leg into your body wall? So I noticed that there was fusion of the proximal leg into the body wall that seemed to be associated with terrestrialization in like, you know, a whole bunch of different groups in arthropods. So it seems like, you know, you have these legs that ancestrally, you know, you doing swimming with, and you're just, you know, you're in the water. But then when you take those legs and you move onto land, you need to like really, you need to make the leg robust in order to like support the body weight on your legs on land. And so I'm thinking that maybe, you know, that's why you would kind of fuse these proximal leg segments into the body wall, because like you're like broadening them instead of making just like a little tiny stick cylinder leg. You know, you're taking those leg segments and you're broadening them in order to make a nice base in order to like support your weight on land. I hope that was helpful. but I would love to look at, like the actual cellular basis of like what you do with this leg.
Okay, we have a few more questions and maybe a minute or two to cover them. Gulherm Gannett says, Heather, how would you apply this perspective of co-option versus dormant fields for structures that are not novel, but rather a repeatedly evolved trait? For example, eyes evolving in different animals with similar genes, or antennules on the tarsi of arthropoda.
I guess I would say, so I think for the eyes example, I think there's good evidence that, we shouldn't just say something is, two traits are homologous or something. We should kind of say, well, at what level are they homologous? So like for eyes, my understanding, maybe it's wrong, is that like all the ancestor of all bilaterians had some kind of, photoreceptive like pigments and stuff that could function as sort of an eye. And then when the groups diverged into like protostomes and deuterostomes and all that stuff, then the eye became like more complicated. So it's like you have the same like basic regulatory architecture like Pax and Alys and stuff, but then you've done different things with them. So it's like they're not homologous as like a camera eye versus like an ommatidia eye, but they are homologous as like a photoreceptor kind of cell patch. And then for the tarsi of, like house centipedes versus insects and stuff. I mean, they're not true leg segments, but they are, they do share the same regulatory stuff. You could maybe express odd skipped, and you wouldn't have like muscles attaching there to make it a true leg segment, but yeah, you could maybe just kind of re-express part of the joint. Yeah, I don't know. I don't know if that's a good question, but
unfortunately, we're out of time for questions. I'm gonna dismiss these to the chat bar, and maybe Heather can get back to you if you stay on the chat.
I will.
And we're gonna move on to our next speaker.
Hi, thanks, Alys. So it's my pleasure to introduce Dr. Eyal Karzbrun. Eyal has a really interesting educational background, primarily in physics. So he did his bachelor's in physics at the Hebrew University, and then went on to the Weizmann Institute, where he first did his master's in theoretical physics, where he studied string theory, and then did his PhD in physics, studying polymer physics of DNA, and figuring out how to assemble artificial cells with basic building blocks. And now more recently, Eyal has been at the UC Santa Barbara and the Kavli Institute for Theoretical Physics. And he's holding the prestigious Human Frontier Science Program Fellowship and the SAGE Fellowship. And today, I'm really excited to hear him talk about how he's taken that background in physics and now started really applying it to some new and interesting biological questions. So with that, take it away, Al.
Thank you very much, Ryan, for this introduction. Let me share the screen. Hopefully it runs smoothly. Can you see the screen?
Yes, sir.
Yeah, okay. So I'm super excited to be here, and I'd like to thank the organizers for the opportunity. My name is Eyal Karzbrun, and I'm a postdoc at the University of California, Santa Barbara, with Sebastian Streichen and Boris Schreiman. And today I'll introduce a new model system which we developed for studying neural tube morphogenesis using human stem cells. I'll begin by briefly saying that the question that motivates our work is whether physics can be used to understand organ development, and whether phenomena that has been studied by physicists in non-living systems has any relevance to organ morphogenesis. So for example, do the wrinkling patterns which appear on the surface of swelling gels have anything to do with gerification patterns in the human cortex? And to address this type of questions, we need quantitative experimental systems that are often hard to realize in a living embryo. And my goal over the last years has been to develop such quantitative systems using human stem cells. And today I'll focus on the neural tube folding morphogenesis. So why should we care about the neural tube? Neural tube formation is one of the earliest morphogenetic events in human development, and it's the first step in the development of the entire nervous system. The image in the slide shows a neural tube from a human embryo at around day 22. At this time point, the neural tube is completely open, and you can see the two opposing neural folds very, very clearly. In healthy embryos, these folds will approach each other and form a closed tube by day 30. However, 1 in 1,000 pregnancies results in neural tube defects in which the neural tube fails to properly close. And although neural tube defects can be studied in animal models, it turns out that there are many human-specific alleles or mutations that are not recapitulated well in mice. And so we simply do not know what those human-specific mutations are doing. And this motivates us to have a new model system, which is based on human genetics for studying neural tube morphogenesis. Beyond the human-specific aspects of neural tube folding, we also don't have a full mechanical understanding of neural tube closure. There's been instrumental work in animal models which deciphered multiple cellular behaviors that are involved in neural tube folding, and these include apical constriction, planar cell polarity, and cell migration. However, because there are so many processes working at the same time, we still don't understand which tissue produces the forces that actually drive the folding. So one hypothesis is that folding is driven entirely by the neural plate as it contracts on the apical side. Another hypothesis is that lateral expansion of the non-neural ectoderm pushes the neural tissue inwards. And yet another hypothesis is that mesoderm expansion underneath the neural plate drives the elevation of the neural folds. And what I would like to argue today is that an in vitro model system can help us distinguish between these hypotheses by first decoupling some of the underlying processes, and also by directly measuring cell behaviors, which is challenging to do in vivo. So how can we construct an in vitro model that captures human neural tube morphogenesis? 3D stem cell cultures or organoids are very promising because they're accessible for imaging and manipulations, and they also allow us to study human genetics. But organoids are not ideal for studying morphogenesis because their shape is very irregular and uncontrollable. Just as an example, you can see images of 2 forebrain organoids that have identical genetic background. They're derived from the same stem cell line by the same poor student, but their shape is completely different. So how can we study morphogenesis, the development of shape, if shape is poorly defined or highly variable in organoids? And what I would like to present to you today is a solution to this problem. It's a new type of a human stem cell culture, which is controlled, reproducible, and experimentally accessible. And most importantly, our approach enables us to study neural folding morphogenesis in vitro. In the next slides, I'll describe our approach, our protocol. I'll try to convince you that stem cells can self-organize into a neural tube, much like the in vivo scenario. And I'll share with you some exciting insights into the physics of neural folding. I'll begin by describing the experimental setup. The key ingredient is setting up a very controlled 3-dimensional initial condition, and for this we use micropatterning. The idea behind this is that reproducible initial conditions will drive reproducible morphogenesis. The micropattern is shown in the illustration in an orange color, and it's composed of adhesive proteins that are deposited locally on a glass surface. When we see stem cells, human stem cells, on the glass surface, we obtain a 2D culture that is confined to the micropattern. It's pluripotent, and it also is an epithelia. You see this, the tight junctions here. Next, we expose the 2D culture to a low concentration of matrigel, and this triggers a robust transition from a 2D epithelia into a 3D tissue containing a single large lumen. I don't show this here, but we verify that pluripotency markers such as OX4, SOX2, and ANOG are expressed in the 3D tissue. So what we achieved is a 3D tissue that can be easily shaped in the lab using micropatterning and has the potential to transform into any organ of the human body because it's made of pluripotent stem cells. And when we expose those stem cells, that 3-dimensional tissue of stem cells, to a cocktail of small molecules or morphogens that are involved in neural induction, we observe a sequence of pattern formation and folding morphogenesis, which are completely self-organized. This is a section of the sample at day 0, before we expose it to the morphogens. A day after the exposure to morphogens, a neural region appears at the center of the culture, which you can see by N-cadherin staining shown in red. And the rest of the tissue, including the dome, expresses the non-neural epithelial marker E-cadherin, which is shown in cyan. At day 2, we observe that the edges of the neural tissue begin to bend upwards, you can notice that the surface ectoderm is now much thinner than the neural tissue. Scale bar is 50 micron. So the neural tissue here is about 200 micron, and it's similar to the dimension of a human neural plate, which is 0.5 millimeter. At day 3, we see that the neural tissue folds inwards, and the ectoderm, again in cyan, curls over it. And finally, on day 4, we observe full closure of the neural tissue and coverage by the non-neural ectoderm. And the sequence of events which we observe, namely pattern formation of neural induction, bending, folding, and closure, are hallmarks of neural tube formation in vivo. And I would like to emphasize that this process is completely self-organized. All we did was set up a very controlled 3-dimensional initial condition, expose the cells to a homogeneous cocktail of small molecules, and which is essentially setting new biochemical boundary conditions. And then the cells did everything by themselves. So it's purely self-organized by cells and their interactions and their response to the morphogens. I would like to go over several additional points of similarity between the in vitro model and in vivo neural tube. On the left, you see a vertical section of the stem cell system, and on the right, you see a section from a chick neural tube. The first common feature between them is the formation of hinge points. These are very small regions in which the neural tissue strongly contracts, and the formation of hinge points is essential for neural tube folding in vivo. The second morphological similarity is the formation of a tissue bilayer of neural and non-neural ectoderm tissues. You can also notice the difference in thickness between the pseudostratified neural tissue and the thin ectoderm layer. And the same features are observed in vivo, as you can see here in this section. Another point of comparison to in vivo neural tube formation is closure of the neural tube. The images in the slide show sequential cross sections that were taken at different positions at the same sample. The leftmost section is positioned in a region where the neural tube is open, and at the rightmost section is positioned at a region in which the neural fold is closed. And the distance here is indicated in microns. And in the transition regions, you can notice that the non-neural epithelia is making contact prior to the closure of the neural tissue. So this indicates that the fusion is mediated by the non-neural ectoderm, and similar observations have been made in mice neural tube in which epidermal protrusions, philopodia, are making contact prior to neural tissue fusion. And just in case there is any doubt, I would like to emphasize that the neural fold completely closes and it's wrapped by a surface ectoderm, again, as the in vivo scenario. Beyond the morphological similarity, we have also verified that the cell type in our system match the key cell type involved in neural tube morphogenesis. The neural tissue expresses PAX6 and OTX2. These are 4 brain markers. And that's important because usually when we say neural tube, people immediately think spinal cord, but what we have is anterior tissue. So we should be thinking brain fold, early brain development. The neural tissue also carries primary cilia, shown here in yellow, that are typical of neuroepithelial cells. The non-neural ectoderm, shown in cyan, expresses transcription factor TFAP2 alpha and keratin 8. These are both markers of surface ectoderm and indicate cells are differentiated towards being epidermal cells. Interestingly, we see a third population of cells that is shown here in green, and this is at the interface of neural and non-neural ectoderm. And these cells express neural crest markers such as SOX10 and neural plate border markers such as PAX7. They also express markers such as snail and slag that indicate that they're undergoing EMT. So those green cells are expressing markers of neural crest cells that are undergoing EMT, which is similar to their in vivo behavior. You know what I would like to emphasize, not only we see the right cell type in the culture, but they're positioned in 3D match their organization in vivo. I would like to note that the system is highly reproducible, and you observe the folding process at about 90% success rate. And you can see a few replicas here on the slide. So to summarize the first part of the talk, I've shown you a human stem cell system that mimics neural tube morphogenesis in terms of morphology, cell fate dynamic, and reproducibility. And now I'd like to go back to the original question of what drives neural folding and how should we understand it from a physical perspective. Our data indicates towards a new mechanism for folding, which I would like to describe in the next slides. And this mechanism is essentially based on surface interactions between neural and ectoderm tissues. And we like to think about this as like a liquid wetting phenomena, like a capillary action. We propose that just like capillary action, which drives liquid up against gravity due to surface interactions between liquid and glass. So in the same fashion, tissue wetting between neural and ectoderm tissues can produce forces that elevates the neural folds upwards and creates folding of the neural tissue. And in the next slides, I'm going to show you 3 experimental indications which support this hypothesis. So the first evidence is that there is a big change in contact angle before folding onset. And before we go into the data, I'll just briefly explain that contact angle is to measure interaction in liquids and surface in material science. And typically a contact angle that is formed between a liquid drop and a surface that is smaller than 90 degrees indicates that the interactions between the liquids and the surface are strong, and we call this a wetting regime. The liquid wants to be in contact with the surface. On contrary, angles that are bigger than 90 degrees indicates that the liquids prefer to be in contact with itself and to minimize the contact with the surface, and we call this a non-wetting regime. Now we apply this concept of contact angle to our experiment, and we look at the angle that is defined at the interface between the neural tissue, the non-neural ectoderm, and the glass. And you see this interface here. This is the neural tissue, the non-neural tissue, and the glass here before. What you can see in the images is that the contact angle transition between a wetting regime of 24 hours, where the angle is smaller than 90 degrees, to a non-wetting regime of 36 hours, where the angle is bigger than 90 degrees. And this change in contact angle occurs before there is any folding of the neural tissue. So folding only happens at around 48 hours. And we quantify this here in the graph below. We measure the contact angle as a function of time in black, and the neural curvature, which is a measure of folding, is shown in red. And you can see that there is a sharp increase in contact angle that precedes the increase in folding. This demonstrates that there is a wetting transition before there is a tissue folding. So again, why should we care about the contact angle? Because like drops of liquids on surfaces, to observe change in contact angle between the tissue suggests that as differentiation takes place, the surface ectoderm and neural tissue increase their affinity towards each other, and they prefer to be in contact with each other rather than being in contact with the glass. And this can provide a force to elevate the neural folds and fold the neural tissue. The second piece of evidence that supports this hypothesis is that we observe high levels of fibronectin at the interface of neural and non-neural tissues. You can see clearly in the images, NCAD labels the neural region in red, and fibronectin is shown in green. In the top, you see a vertical section of the fold. This is a neural region, and that's fibronectin at the edge of the neural region. Here you see a horizontal section. Again, this is the neural tissue, and there is strong fibronectin at the edge of the neural tissue. And when we look at the interface between neural and the glass, we almost don't see any fibronectin signal here. And again, this is quantified here. You see fibronectin at the neural, non-neural interface is much stronger than fibronectin at the glass. And this differential ECM composition could be the mechanism which explains why neural and ectoderm tissues prefer to be in contact with each other rather than being in contact with glass. I'll also briefly say that this differential ECM composition has also been observed in mice and chick neural tube closure, where different parts of the neural tissues are covered with different compositions of extracellular matrix proteins. And we were very surprised to see fibronectin in these tissues because we don't add any fibronectin to the culture. So this implies that fibronectin is somehow synthesized by the cells during the morphogenetic process and also localized to that interface by the cells. We did single cell sequencing, and that revealed that the non-neural ectoderm, the surface ectoderm, is actually the cell type that the cell that produces the fibronectin, and this suggests a human-specific role for the non-neural ectoderm in folding morphogenesis, because, for example, in mice, fibronectin is not produced by the ectoderm, it's produced by mesoderm. Okay, so I've shown you so far two indications for a sort of a wetting phenomena that drives folding. First, the contact angle, then this differential ECM composition. Finally, I want to show you a third evidence that supports this mechanism, and this is when we inhibit, when we do an experiment to inhibit fibronectin matrix, we don't observe an interface, and we also don't observe folding of the neural tissue. So what you see here are three samples. The top is a control sample. You can clearly see the folding neural tissue. You can see the hinge points. You can see the interface between neural and non-neural is indicated by the yellow line. And when in the second sample, we use a small molecule novobiotine to inhibit fibronectin matrix. And you can see that the neural tissue is completely flat, and the interface between neural and surface ectoderm is practically abolished. The last sample was treated with ROCK inhibitor to reduce contractility. And we observed that loss of contractility also results in folding defects. There is no folding of the neural tissue, but we still see some interface between the neural and non-neural ectoderm. And this suggests that there are two independent processes that are going on here. One is interface formation, which depends on fibronectin matrix assembly. And the second is neural contractility. So to convince ourselves that our hypothesis is in the right direction, we made a small computational model that takes into account only those two cellular activities. The first is that neural and ectoderm prefer to be in contact with each other, and this is what we call the wetting parameter. And the second is that neural tissue contracts on the apical side. And our models show that these two behaviors are sufficient to reproduce the in vitro experiments. And we see that wetting is essential for the formation of a neural and ectoderm bilayer, and that contractility is required to further pull those neural folds inwards and to close the neural tube. And you can see that without wetting, the neural tissue is flat and there is no interface formation. Without contractility, we do see neural folds, we do see an interface, but those folds are open. So we need both parameters in order to see the morphology of the full folding neural tube. And what's remarkable is that the morphologies which we observe in this very naive model are quite similar to the experimental observations. In particular, we observe a thick neural tissue and hinge point formations, which are both emergent properties of the model and were not put in by hand. And this is very interesting because the paradigm is that hinge points form because there are some local cell behaviors at the hinge regions which drive the hinge to form. So for example, there is one hypothesis that there is excess contractility here. But in the model, we observe hinge formation, although the cellular behaviors are homogeneous. All the cells, all the neural cells are doing the same thing. So one more thing that we see is that the model also predicts how the neural fold shape changes as we increase the size of the sample. We observe that for narrow micropatterns in the experiment, a single hinge, but for wider micropatterns, we observe two lateral hinges, and the same behavior is recapitulated by the model. And this consistency between model and experiment further supports the hypothesis that tissue wetting is an important factor in neural folding morphogenesis. So to quickly summarize, I've shown you today a minimal and quantitative stem cell system that self-organizes into a neural tube. The take-home message here is that controlling initial conditions of organoid formation is key to having reproducible morphogenesis. In our case, we achieve this by micropatterning, but one can envision other ways to do so. And finally, this system enables us to propose a new physical mechanism that may be the driving force behind neural folding, and there's still work here to actually prove that this is the correct mechanism and that it has relevance in vivo. I would like to conclude now by thanking my advisors, Sebastian Streichen and Boris Schreiman. I would also like to acknowledge Amel Kamkel, a graduate student in Sebastian Lab, and Heto Megali, who have been working with me intensively on this project. Noah Mitchell contributed codes for 3D representation and analysis of the neural tube. Stella Glauser and Kenny Konsik helped us with single-cell sequencing and analysis. And we also benefited a lot from collaborations with Eric Sidia and George Britton and Arya Warflash. So thank you for listening, and I'm happy to take any questions.
Thanks, Eyal. That's really amazing system. So we have some questions. Catherine Rogers asks, I assume morphogens are added uniformly to culture medium, but is there any indication that the resulting signaling is actually asymmetric? And she's trying to understand how spatial expression of neural tube genes is preserved, since it's thought to result from asymmetric signaling distribution in vivo.
I'm not sure to which asymmetry Catherine is referring to, but I'll say what I know about our system. So we add BMP is one of the driving signals that triggers neural induction or ectoderm differentiation in vivo, and we use BMP in the system. And we add it homogeneously. But previous works, especially by Warmflash and Sidia, have shown that when you use cells on micropattern, although you expose the cells homogeneously to say BMP, it doesn't penetrate homogeneously into the culture. and therefore effectively you get a gradient in the culture. So cells that are deep in the culture don't sense BMP, say, and cells that are on the periphery do sense BMP. And this triggers the first symmetry breaking. Now, neural tube development would also incorporate anterior-posterior symmetry breaking and dorsoventral symmetry breaking, but we don't have this here. From single-cell data, it seems that all of the neural tissue that we have would match, say, dorsal anterior neural folds. So to have a full neural tube anatomy, including DV pattern formation, we would need to include some ventral source, ventral signaling source, for example, sonic hedgehog, which would be a next step, I guess.
All right, Mohammed Simsek asks, what drives outer cells to express ECAD, but central ones to express NCAD after being exposed to the same morphogen cocktail? Did the cells in the center transition from expressing ECAD to NCAD?
So initially, so I think it's related questions. So like I said, although we put BMP homogeneously, the penetration of BMP is not homogeneous. So different cells feel effective different levels of BMP. Initially, the cells, if we don't add BMP, the cells would all express NCAD. So that's the default state of those cells to go into neural fate. So you need to add BMP in order to drive ectoderm, non-neural ectoderm expression. I would say that an important factor in our system is that we start with the 3D configuration with a lumen, and we check that if we don't have that, and we start just with 2D, we observe the cell fates, but we don't observe the morphogenesis. We don't observe the folding morphogenesis. And one hypothesis is that there is some signaling that occurs within that lumen that is important for folding, which we have not yet resolved. When we looked at single cell data, we do see that cells themselves express BMP inhibitors, they express WNTs. So there is some intracellular signaling that goes on beyond sort of the boundary conditions that we impose on the culture.
Interesting. Martin Aristegui asks, are the neural crests SOX10 positive cells migratory in the system? And basically, this could be a promising system to study neural crest delamination and migration.
Yes, absolutely. So we haven't resolved exactly the time points at which we appear. We see early markers of plate border, which appear before those cells migrate. And then, like I said, we also see SLAG and SLAG, which indicate that they're undergoing EMT. There is a live reporter, a SOX10 live reporter, which we use, and we see those cells are actually moving around. And when you look at them in 3D, it looks like there is some stream kind of configuration to them. So those are all indications that they're undergoing migratory behavior like they're undergoing in vivo. And I agree, that's a very promising direction to take forward.
All right, last question from Flavio Zalesi. Great talk. Have you considered attraction force between the non-neural and neural ectoderm explaining the elevation of the folds?
Yeah, so I think what I've shown you so far is sort of like a tissue level analysis of the wetting and ECM compositions, which indicates towards some force generated by the interaction between the two tissues. And I think to really resolve that, we need to go in and really measure piconewton forces that those cells apply. So either doing an assay with adhesion to actually measure what's the adhesion between those cell types, and then going with traction force microscopy and see how much force they apply on the surface, and also supplementing that with live imaging to see cells really go into the interface and so on. So I think that it will take some time until we can really resolve where are the forces that drive folding are coming from, and I think that's a very interesting direction to go into.
Okay, I think we have to cut it off there. So I just want to thank both Dr. Bruce and Dr. Karzbrun for their awesome talks. I'll remind everyone that the seminars are recorded, and those should be online on Monday. And with that, I just reintroduce Dr. Whitney Edwards for a quick announcement.
Hi, everyone. Thank you again to our speakers today. I just want to announce the remaining postdocs that will be presenting for this year's seminar. So we have selected Chi-Chung Wu, Isabel Espinosa Medina, Maria Sally, Fethullah Simsek, Miguel Salinas Saavedra, Sudha Rajdurkar, Jennifer McKee, Frank Macabenta, Joey Gersi, Elle Robertson, Alys Okorzi, Jacqueline Chen, Joshua Abrams, Catherine Criswell, Margarita Perillo, and Jason Sinclair. So congratulations to all of the postdocs that were selected this year. And we're really excited for you all to join us again at next month's seminar, October 8th, to hear Chi-Chung Wu from the Max Planck Institute for Heart and Lung Research, and also Isabella Espinosa Medina from Howard Hughes Medical Institute at Genelia Research Campus. And so thank you all for coming.